We are on a mission to stop an evil scientist. He has discovered a 4-amino acid protein that he can use to destroy the world. Our plan is to break into his lab and mutate his sequence so the protein will no longer be dangerous. Okay, so we're in, and I believe that is his DNA sequence laying on the bench. Oh no, we're going to need to identify the reading frame. But this is just his DNA sequence, and after we transcribe it, all of the thymines will become uracils. This is the transcribed mRNA sequence. Oh, and luckily I brought my amino acid chart. The reading frame begins with a start codon, which is always AUG. Once you find your start codon, you can translate the sequence one codon at a time until you reach a stop codon, which does not code for an amino acid. There are 3 different stop codons, which are all identified on the chart in yellow. All mRNA outside of the reading frame will not be translated. Okay, I think I've found the reading frame and I translated the strand for you. Now let's think about mutations. We will use a base substitution to cause the mutation for our mission so that we can do it quickly before he finds out we're here. 2 different types of substitutions can occur as the bases can be split into groups by their structures. Adenine and guanine are purines, and cytosine, uracil, and thymine are pyrimidines. If the bases involved in the substitution both have the same structure, the substitution is known as a transition. An example of this would be swapping an A with a G. A transversion occurs when a base is substituted by another base with a different structure, like if an A was substituted by a C. Now I'll quickly brief you on the 4 mutations that might help us alter the protein's function. In option 1, a transition would occur as the highlighted T in the original DNA sequence would be changed to a C. When we translate the sequence, the second amino acid will become alanine. This type of mutation is known as a missense mutation, and it results in a different amino acid being incorporated into the protein. However, note that for this mutation, the mutated codon must code for an amino acid. It cannot become a stop codon. In option 2, a transversion would occur as the highlighted A in the original DNA sequence would be changed to a T. When we translate the sequence, the 4th codon becomes a stop codon and results in premature termination of translation, a shorter amino acid sequence, and a shorter protein. This is known as a nonsense mutation, and it will most likely result in a non-functional protein, particularly if the mutation occurs early in the sequence. In option 3, a transition would occur as the highlighted C was substituted with a T. However, when we translate, there is no change in the amino acid sequence. This is known as a silent mutation. You can think of it like when you do the work for a math problem wrong but somehow end up with the right answer. Even though the sequence does not change, these mutations can have phenotypic effects on the protein. Some examples of the effects they can cause are affecting the amount of protein in a cell, affecting the folding of the protein, affecting the mRNA splicing, and affecting the binding of miRNAs. In this final option, a transversion occurred as the highlighted A in our original DNA sequence was substituted with the T. When we translate the sequence, the former stop codon is changed into a codon that does code for an amino acid. This is known as a read-through mutation and results in a longer amino acid and protein, as we must now continue translating the sequence until we reach the next stop codon. Okay, so which mutation do you think will work best to foil the plan? Yes, I agree. We should use the nonsense mutation so that we can be fairly certain that the mutated protein will be non-functional, which will allow us to stop the evil scientist. Thank you for your help.